Bismillahirrahmanirrahim. Assalamu alaikum. Today we will study the form elements of the blood, and among them the most important is the RBC. So today's lecture consists of the production of RBC, description of RBC, their function, how they are formed, and in the body where their genesis occur. All about the RBC. Today we will study that one. About the red blood cells, the most important thing regarding their function is the transport function of RBC. The other name for the RBC is erythrocytes. The first and most important function of the RBC is to transport the hemoglobin, and it in turn carries oxygen from the lungs to the tissues. As our every tissue, every cell needs oxygen, so to provide the oxygen to the cells, the RBC or the blood and RBC, it is the major source through which the transport of oxygen take place. RBC have other function beside transport of hemoglobin as well. They contain a large quantity of a very specific enzyme which is named as carbonic anhydrase. This specific enzyme in our body present at certain specific places, and this may be asked in viva, at which the question viva question is at which places the carbonic anhydrase present in our body. So one of them is the RBC, other is the GIT, kidney, brain. So these are some of the specific places and also the lungs. Due to the presence of this specific carbonic anhydrase enzyme, very specific reaction take place through which the reversible reaction between the carbon dioxide and water to form the carbonic acid that take place. So the repetitive of this reaction makes it possible for the water of the blood to transport abundant amount of carbon dioxide in the form of bicarbonate ion from the tissues to the lungs, where it is reconverted to carbon dioxide and expelled into the atmosphere as a body waste product. The hemoglobin in the cell is an excellent acid-base buffer. Regarding the function of the carbonic anhydrase regarding the oxygen transport, that will be detailed and discussed in detail while we will discuss the respiratory unit. Okay, the next thing, the shape and the size of the RBC. Regarding the normal red blood cells, they are normally biconcave discs having a mean diameter of 7.8 micrometers and a thickness of 2.5 micrometers. The thickest point is 2.5 micrometer, while surrounding the center 1 micrometer is the thickness. In healthy men, the average number of RBC per cubic millimeter is 5200,000, while in women that is the 4700,000. Again, this may be asked in viva, why The number of RBC are more in men as compared to the adult women. And the reason behind that is, the answer of this question is due to the menstrual cycle. In the woman, in adult woman, the number of RBC is lesser than the adult male. Red blood cells have ability to concentrate hemoglobin in the fluid up to about 34 grams in each 100 milliliters of the cells. Now comes the production of the RBC, how they are produced and where. The most important thing is where in the body they are produced. So the areas of the body that produce RBCs that involve different places at different our developmental stages. In the early weeks of embryonic life, primitive nucleated RBCs are produced in the yolk sac. During the middle trimester of gestation, the liver is the main organ for the production of the RBC, but reasonable numbers are also produced in the spleen and lymph nodes. Then, during the last month or so of gestation and afterward, RBC are produced mainly in the bone marrow. And through this graph, we can see the age in years below, and this is the percent, cellularity percent. As I have said you, in the last years, that is after birth, the production of RBC is mainly in the bone marrow. So because of that, the tibia, shaft, femur and all these bones, they are shown here, in which the bone marrow of which these bones, the RBC that is produced. Now comes the genesis of the blood cells, different developmental stages. The red blood cells begin their lives in the bone marrow from a single type of the cell, which is called the pluripotential hematopoietic stem cell, from which all the cells of the circulating blood are eventually derived. As these cells reproduce, a small portion of them remains exactly like the original pluripotent cell and is retained in the bone marrow to maintain a supply of these, although their number diminish with the age. So you can see this is the main pluripotent hematopoietic stem cell. From this cell, all the other types of the cell, they are generated. But you must remember that some of the original cell that remains there to produce further different type of the blood cells. Specifically for the production of the RBC, these are the different stages for the development of the erythrocytes or RBC. To remember these stages, Proerythroblast, basophil erythroblast, polychromatophil erythroblast, orthochromatic, reticulocyte and mature RBC. To remember the sequence of developmental stages, I tell you a specific word, that is probe, P-R-O-B, probe, pore, P-O-R-E. Probe pore means probe is a surgical instrument through which we can drain the wound, we can clean an injured site, and pore means just the hole. So just to remember the sequence of developmental stages of RBC, you can remember this word, that is probe pore. that is the whole of their specific surgical instrument. The first is the proerythroblast, then basophil erythroblast, polychromatophil erythroblast, orthochromatic and reticulocyte, and then mature erythrocytes. This is the life of a red blood cell, how it matures. On one hand, I am showing the changes which grossly occur during the development of the RBC, and here I am showing how this looks like from inside. So the first stage is the proerythroblast. From there we have the basophilic erythroblast, in which basophilic cytoplasm with the perinuclear halo is present. Sometimes the cytoplasm bulges from its oval counter to form a small ear-shaped process. Nuclear chromatin is not homogeneous, and you can see here nucleolus as well. The next is the basophilic erythroblast, having the diameter smaller than the proerythroblast. Nuclear chromatin shows a strong contrast between the light-colored areas and dark violet chromatin particles. The cytoplasm is moderately basophilic, reflecting high protein and RNA content. Polychromatophilic erythroblast, it has a mature nucleus with nuclear condensation. Its cytoplasm has a grey hue derived from the hemoglobin. No, you must note that the nuclear to the cytoplasm ratio also. The next stage is the orthochromatic erythroblast, which is the last erythroid precursor with the nucleus. The nucleus is compact and situated near the membrane prior to its expulsion from the cell. The cytoplasm is similar to that of a mature RBC, reflecting a high hemoglobin content. The next thing is the reticulocyte, which is the young erythrocyte with granular or reticular filamentous structure constituting 0.5 to 2% of all erythrocytes. This reticulum network is only visible using vital staining. Reticulocytosis is frequently occurs after hemolysis or acute blood loss. Then we have the mature erythrocytes, which is circular, reddish, biconcave, having the size 7.8 microns and no visible internal structure. The hemoglobin content in these cells makes their typical pink-red color. The brightness in the center of the erythrocyte is caused by the biconcave shape. A very important thing about the mature RBC is that it is without nucleus, and this question that may be asked during the viva, that uh, through two ways we can ask the question about the nucleus of the RBC. Number one way is that I can ask you the size of the nucleus of mature RBC. I am giving you this assignment and you must tell the answer of this one. What is the size of the nucleus in the mature RBC? And second question may be asked, that is that if you say that the mature RBC is without nucleus, so how it survives? So answer to this question I will tell you in the next lecture, but here I am giving you the assignment. You must search the answer of these two questions. Regulation of RBC production and having the role of erythropoietin. What is that one and how it is going to help and affect the genesis of the RBC? Any condition that causes the quantity of oxygen transported to the tissues to decrease, that is the hypoxic condition. Hypoxic condition is in which the tissue oxygenation is defective, is lesser than the normal. In that case, it is a strong stimulant for the production of the or the genesis of the RBC. And for that one, the erythropoietin, which is a very specific enzyme released by the kidney because of the effect of hypoxic condition. In response to the hypoxia, kidney releases erythropoietin and that reaches to the bone marrow to stimulate increased production of RBC. But you must see this picture also, that are factors that decrease oxygenation. Some of the factor: low blood volume, anemia, low hemoglobin, poor blood flow, coronary disease, hypoxia. All these conditions, they are going to create the condition tissue oxygenation decrease, and that condition is called hypoxic condition. So mainly the hypoxia that is going to affect or to increase the genesis of the RBC. But how? The hypoxia affects the kidney. In response to the hypoxia, kidney produces erythropoietin and then it reaches to the bone marrow and it affects the genesis or it increases the genesis of the RBC. Regarding the maturation of the RBC, for the maturation, the basic requirement for RBC is the presence of vitamin B12 and folic acid. Both of these are essential for the synthesis of DNA because each in a different way is required for the formation of thymidine triphosphate, one of the essential building blocks of DNA. Therefore, lack of either vitamin B12 or folic acid causes abnormal and diminished DNA and consequently failure of nuclear maturation and cell division. So with this, we complete the genesis of RBC, introduction to the RBC, genesis of RBC, and how they are affected by the hypoxic condition. With that, we complete our lecture. In the next, we will discuss the other topic related to the RBC generation, RBC and hemoglobin formation. Up to that, thank you and Allah Hafiz.